Dear colleagues, let me introduce the possibilities of diploma thesis at the Faculty of Pharmacy, Masaryk University. As you know, the diploma thesis is a obligatory part of the studies, and therefore each student must finish the studies with a defense of diploma thesis, and later on also the state examination. And the diploma thesis is part of the state examination, and students usually start to work on diploma thesis in the fourth year of studies. Generally, there are two ways how to register or how to obtain a topic for diploma thesis. The first is the active search for the topic of diploma thesis by contacting teachers of the departments of a Faculty of Pharmacy. The second is registration on the topic, which will be later on offered by the faculty, and you can see the topics at the web page in the IS system. I would like to shortly introduce the research topics of Faculty of Pharmacy, because in vast majority of cases, the work of diploma thesis is connected with the research which is carried out at the department, and usually the results of such diploma theses are integrated into scientific projects, which are later on usually presented on scientific conferences or in scientific literature. The uniform covers possibly all disciplines of the pharmaceutical research. So you know that we have several departments, and you can relatively simply connect the name of the department with the research topics. So we work on isolation and identification of new biologically active compounds from plants or other natural materials. We work on synthesis, structure elucidation, and also we study the physicochemical properties of different medicinal substances. Doesn't matter if these substances are new, are isolated from natural material, or are synthesized. Sometimes we study the physicochemical properties and general characteristics of substances which are used in medicine. Based on this, we usually continue with these compounds to testing of their biological activity. Generally, we do the preclinical testing, so we use testing on different tissue cultures, cellular models. We use preclinical testing on laboratory animals, and because later on such as developed compounds should be introduced to therapy, so some colleagues work also on the development of different drug delivery systems, including advanced drug delivery system, for example, for the control release of drug substances. And pharmacy is inevitably connected also with clinical pharmaceutical care and general pharmacy. So we can offer topics also connected, for example, with pharmacoeconomy, the pharmaceutical law, or general pharmaceutical care. Now I would like to move to specific departments. I would like to shortly introduce departments and explain which topics are solved at these workplaces. So the first what I would like to mention is Department of Pharmaceutical Technology, which is generally focused on development of different drug and application forms. You know how the faculty is organized. You know that generally we can say that each department covers a specific field of the pharmaceutical research. You know that this is also connected with the state examinations, and each department is generally responsible for the teaching activities covering the topic. Here we have drug formulation. So the research on department is connected with development and evaluation of, we can say, all types of drug dosage forms. So they focus on solid forms, semi-solid forms, and also liquid dosage forms. And they work on possibilities of utilization of this dosage form, not only in pharmacy, but also in cosmetics, in medicinal devices. They have cooperation with veterinary doctors, and they study the utilization of modern drug forms in veterinary medicine. And of course, they also study the utilization of different application forms in food industry, because nowadays the research in area of active bioactive chemical substances is connected not only with pharmacy, but also with some food supplements, or also medicinal foods. Touching the topics, this is not only about the formulation, but it's also about the evaluation of the quality of material which is produced. So it means stability testing, it means analytical chemistry connected, for example, with high performance liquid chromatography, HPLC. It's also about the release of active substances from different drug forms. So the typical application of this solution. The department is equipped with a relatively big spectrum of machines which can be used for grinding, homogenization, granulation procedures. You can study formulation of tablets, capsules, different fluid processes, and colleagues from the Department of Pharmaceutical Technology work also with 3D printers to prepare the materials, let's say very modern materials for the individual dosage, and again release the processing of materials for the control release of compounds. Some projects which are connected with this development of detection material, detection tubes for different toxic substances. This was, and this still, it's project connected with military industry, because these detection tubes are designed for the identification of potential warfare toxins. The preparation of microparticle systems, and not only preparation of these microparticles, but also their evaluation utilizing electron microscopy. And as I said, general analysis and evaluation of dosage forms, for example, by HPLC or other analytical techniques. The second department which I would like to show you is Department of Natural Drugs. Natural drugs. So we study all aspects of the plant secondary metabolism, connected with potential utilization of these compounds or in medicine, pharmacy, and again, also here, the topic is not limited to pharmacy, but we can study also materials and compounds which could be used in food industry or in cosmetics. The general process of isolation of the plant secondary metabolites is connected with different extraction techniques. We have, again, a relatively modern spectrum of apparatus which can be used for the obtaining of different types of extracts. These extracts are then generally analyzed using different chromatographic techniques, and compounds which are later on isolated by preparative chromatography are identified using mass spectrometry and nuclear magnetic resonance. All compounds which we try to isolate and identify are then tested in different biological studies. At department, we do not do many direct identification of the biological activities, but we have cooperation with, for example, Department of Molecular Pharmacy or Department of Pharmacology and Toxicology, or we have cooperation with other institutes, both in Czech Republic and generally we can say worldwide. The number of plants, we do not have to limit the plant material which we process. We are generally focused on plants from the plant family Moraceae, Fabaceae, maybe Euphorbiaceae, but if there is anybody thinking about interesting plant material which can be processed, we are open to these proposals if this is relevant and if the plant is not well explored by previous studies. We can include it into our program. Generally, we work especially with compounds of this type. So we work with phenolics, with terpenoid side chain. And to show you the possible results of such studies, you can show the spectrum of compounds which we isolated from Morus, mulberry, or from Paulovnia. And you can see that these compounds found the application, especially in could find application, especially in treatment of different inflammations, and they show also antibacterial activity. Touching the topics, our department, at the Department of Natural Drugs, students should not be scared of chemistry, should not be afraid of the laboratory work. Sometimes we say that this is like ant work. You have to process a lot of plant material, you have to prepare a lot of fractions, you must be well skilled in making of the thin layer chromatography. Generally, at the department, you can meet all types of chromatography. And if you are keen in separation techniques and connection of separation techniques with the plant material, you are welcome. The next is Department of Chemical Drugs. Department of Chemical Drugs generally is connected more with synthetic compounds. However, now the Department of Natural Drugs has some cooperation with the Department of Chemical Drugs, and colleagues from the Department of Chemical Drugs are trying to synthesize the substances which we isolated in minute amounts to have the material for further work, especially for the bioactivity bioactivity studies. The work of the Department of Chemical Drugs is very variable. You can see modern synthetic procedures, including development of methodologies for stereoselective synthesis, including, for example, preparation of catalytic reagents. Colleagues work on the synthesis of a homologic series of compounds, which then are used to study the relationship between the chemical structure and the biological activity. The studies at the Department of Chemical Drugs, again, do not cover the synthesis only, but the analytical chemistry, which is used to study physicochemical properties of drugs, and also analysis of chemical interactions with biomolecules. And of course, if they have such as homologic series connected with the bioactivity testing, they use the results for the QSAR designing of new structures for the synthesis. If we will talk about topics of the synthetic studies, they synthesize compounds with antimicrobial activities, some compounds with the antimicrobacterial activity, anti-cancer substances, and also compounds which can be used for the treatment of cardiovascular disorders and also for treatment of neurodegeneration. Again, here, if you would like to register the diploma thesis at the Department of Chemical Drugs, you should not be afraid of chemistry. But on the other hand, according to me, the work is definitely not monothematic. And if you like, you can expand your work and find overlaps, for example, with the testing of bioactivities at other departments of faculty. Department of Pharmacology and Toxicology. I think we can say that there are two branches of research. The first is the testing of the chemical compounds for bioactivity in vitro. And the second one is the testing of the bioactivity in vivo. The in vitro and in vivo testing is closely connected. I will start with the testing of compounds in animal models. The department has permission to work on different laboratory animals. If you can see the work with mice, you can see the work with rats. The biggest animal which can be used at the department are rabbits, but generally the most commonly utilized are rats and mice. And the spectrum of bioactivities is connected especially in the models of inflammation, so testing of potential anti-inflammatory compounds, wound healing activity, and also anti-cancer activity models. And all these things can be also done on cell cultures. Usually the testing in vitro on cell cultures goes on before the transfer of the compounds to animal models, because of course better to save money, save time, and save animals to select the potentially active compounds in cell cultures, and later on only the compounds which are somehow promising in the bioactivity are analyzed in animal models. This testing is connected not only with compounds which are newly synthesized or isolated from natural material and faculty, but these models are used for biological evaluation of, let's say, new medical devices, new medical materials, and this is also connected with so-called translation medicine. The work at the Department of Pharmacology and Toxicology is specific. The work with animal models, of course, you don't have to be afraid of animals. And of course, during such experiments, the animals are sacrificed. At the end of the experiment, the animals are sacrificed. It's very specific work which needs the special training, and usually the students are allowed to participate on the testing, but not directly work with laboratory animals, because for such experiments you need the permission of the ethical commission of the department Ministry of Agriculture. But later on, the results are processed. The processing is also very interesting. Such as processing is also very interesting thing. And what I think this is also connected with utilization of the vast spectrum of statistical models. I think the work on cell cultures is, let's say, more accessible. And again, here, what you need to know, or what you will do, you will work with molecular biology methods. You have to be very precise. And again, here you have to be skilled, at least basically skilled in statistical evaluation of the results which you obtain, because without statistics here you will not get reliable material. Department of Molecular Pharmacy. Department of Molecular Pharmacy was previously part of the Department of Natural Drugs, but now it became a separate department. The activities of Department of Molecular Pharmacy are closely related to the activities of Department of Natural Drugs, and we can possibly say that all compounds which are processed the Department of Natural Drugs later on go to Department of Molecular Pharmacy to be tested, especially in vitro, by molecular biology methods, to understand their antioxidant, anti-inflammatory, and anti-diabetic activity. And also, and this is especially in like a tripartite, the cooperation with Department of Pharmacology and Toxicology, to understand the cytotoxicity of these compounds. A big part of the program of Department of Molecular Pharmacy is testing of antimicrobial activity. And nowadays, it's not only the direct killing of bacteria and analysis of minimal inhibitory concentration of active substances, but also the effects on cellular communications of the different bacterial strains, and also the effect on formation of biofilm, because the intercellular communication and biofilm formation are factors which contribute to virulence and the resistance of microorganisms to antibacterial therapy. So the goal is to understand how the natural compounds which we isolate work on single bacterial cells, and if these compounds could contribute to the solution of the problem of bacterial resistance. Part of the research of Department of Molecular Pharmacy is carried out in cooperation with the Academy of Science, the Institute of Biophysics, and this is connected especially with molecular oncology unit, and they study p53 family of tumor suppressor proteins, and also the interaction of different compounds, not only the compounds from Department of Natural Drugs, but so synthetic compounds with this p53 protein. Colleagues at department work also on phytotoxicity of different pharmaceuticals in wastewater, and they have also close cooperation with the St. Anne Hospital, the unit processing and studying the cultivation of medicinal cannabis. I don't know if it's correct, the last but not least, or the least but not last. No, it's Department of Applied Pharmacy. And the Department of Applied Pharmacy, sometimes it's said that this is not an experimental department. Correct on this is that you will not use the lab coat, you will not go to the laboratory. The research areas at the Department of Applied Pharmacy are more connected with something like data mining. And the key research areas are connected with analysis of the drug consumption, about health literacy in medicines, about general pharmacoeconomics and pharmacoepidemiology, and of course, for example, so legal and ethical aspects of all pharmaceutical activities. Dear colleagues, this is a brief introduction of the research topics at departments of Faculty of Pharmacy. What I forgot to say is that all departments of the faculty have many cooperations, not only within the university, but also in Czech Republic and also in Europe. And in all departments, you can find the topics which can be later on used, for example, for the application in Erasmus Plus program. I hope that this presentation gave you the overview of the research, and I hope that it will at least a little bit help you to select the topic which will give you not only the experience and knowledge, but also at least a little bit of pleasure of the contact with people at the respective department. And later on, maybe the topic for your further studies, and maybe also the continuation of your studies, for example, in doctoral study program. Thank you.